Hi, early stages of animal life. Let's go. So, fossils preserve organisms from the past and their traces and traces of the environment. These are just literal time capsules, and things get altered, and a lot of things get lost, but sometimes more is retained than you would think. So, look at these fossils. They can actually be dissected to answer this first question. The stuff on top is from the outer side, and the stuff on the bottom, if you were to dig through that, you would find the middle layers and the bottom layer. Everything is flattened down. That's what happens with rock layers, but it's all still there. And you have to be real careful, but it is possible to actually read that out and recover some of that information. And so, just looking at a fossil like this, we could look at its stomach contents. We can look at what appears to be hair on its back. We can look at the Raman microscopy signal from that hair and determine if it might have melanin in it, because we know what color melanin should be in modern animals, and that it's pretty common in vertebrates. And so we could tell, okay, this part of the animal was probably covered in black fur. This probably wasn't. There are traces of the original molecules. Even if most of the organism is gone, fossils still preserve quite a lot of information. And that's a thing people have been learning just recently. And look at this Leptosidium. Look how ridiculous it looks. A little elephant like nose running on two giant legs. Absolutely off the chain. Here we go. Fossils sometimes preserve very delicate structures. These are all brachiopods, which is a kind of, it's basically a worm with an elaborate feeding apparatus, two arms that are sometimes curled around. You can see the curls right over here. See all these little spirals? That's the mineralized support where the arms would have attached. And you can see it hooks up to the back of the shell right here. This is a food conveyor belt. They're capturing the food as they filter water, and they are sending it right back here along there, and that's where the mouth would have been. So, nonstop food conveyor belt sounds pretty good. Over here, we have some fossils that still preserve some of their original color. See the little spots on these. They can bleach out. They often do bleach out. But when the color is retained in those rare cases, it often doesn't have the same appearance that it originally would have. But if you can find the chemicals that the organism was excreting to make the color, you can usually tell what those are even after fossilization. You can say, oh, this is derived from a pigment that would have been red originally when it was alive, for example. So, what do we need in order to preserve color? Basically, no sunlight and really lucky conditions. Certain types of fossilization, this happens, but it is very rare that you get any color information because you have to have preservation of organic material, which is what the molecules are that are giving it the color. If that gets too heavily altered or too heavily bleached before preservation or after fossilization at any point during those millions of years, then yeah, the color is very fugitive. It is easily lost. Okay, how can we connect extinct and extant, that means still living organisms? We we can do it through biomolecules, like the molecules that make the color, like the molecules that make up cholesterol in cells. Very useful. Those will often be preserved in and around the fossil of an animal, for example. And you can extract them and you can actually demonstrate what sort of creature that was sometimes. Oh, and this is a little brachiopod baby up here. Yeah, just because we like brachiopods. We can also make phylogenetic trees, like little family trees, but we can do it instead of using nucleotide or protein information, we can use information on body parts. We can say, does it have a big spiral on both sides? And so that's a 1 if it does, a 0 if it doesn't. Like these guys do not have that sort of spiral inside, and so on. Does it have little reddish brown spots on it when it was alive? Yes or no? And if you get enough of those characters, and if they're the right sorts of characters that have something to do with the genetics of an organism today, like major changes, then yeah, you can actually use those to classify fossils. And it appears to make sense. All right. How might we locate the birth canal of an ichthyosaur? Yeah, just a bunch of bones up here. You say, okay, if this was a person, what have we got? That's an analog of the head. Okay. And these are analogs of the arms. And yeah, there's one bone that makes up the upper arm. Oh, sorry, it's right here. That's a shoulder bone. And then it splits and it's got two bones right here. That's like your bone up here splitting, and then you got your two bones right here. Yeah. They're basically the same creature, and so you'll see this one has, it looks like four major fingers running down there with more little phalanges in them. But yeah, that's a fairly insubstantial change. So then look back here, there's a back leg, and you say, oh, it's probably between the back legs, and sure enough, that's where the baby ichthyosaur is coming out. Now, this mother and baby died in the process of giving birth or very soon after, and we don't know what killed them for sure, but yeah, maybe the baby got stuck there. Yeah, hard to say. But this suggests very strongly there's no other good reason for this to be positioned in between the legs, basically, of a larger member of the same species. That is probably the mother, that is probably her baby, and they probably died at the same time related to the process of giving birth. Or there's one theory that the baby was maybe ejected from the womb after the mother died while heavily pregnant. It's possible too, yeah. So yeah, we can use our best Sherlock Holmes logical inference and all the data available to us to at least try to come up with a plausible cause of death. There are no right or wrong answers, let's say that, in paleontology, but there are answers that use all of the information very well, and there are answers that ignore giant parts of the information, and you might as well call those wrong because they're pretty bad. So, yeah. Okay. We can differentiate sometimes muscle and fat and cartilage and all sorts of things. So here is an animal. It's got a little, I don't know if you can see that little, it's like a little line running there. We think that's actually the remnants of its notochord. The notochord is not preserved here, but we have an impression of it, and we have the altered remains that may have come from it. But then you also see how it's divided into these blocks, and those look exactly like what you get when you put a fish out in the sun. It's got muscles. The muscles are in these great big blocks attached together, and as it dies and the organism dries out after it's died, you will get a little bit of contraction of those. So remember that fossils are always things that have died. Now they can get buried alive and die kind of after burial. Those make really great fossils, but kind of horrifying. Or yeah, they can die and then fall down and then just get really, really lucky, land in the right spot, and get preserved forever. This is a Tully monstrum, also known as the Tully monster. Yeah. We think it's got little eyes on this, they call it the bar organ. It's basically a hardened support that holds the eyes out away from the body. And I don't know, people argue back and forth about what this thing is and why it looks so weird compared to things that exist now. This is only around 300 to 330, somewhere in there, million years old. So, yeah. Actually not that long in geologic time, and there are a lot of things from that time that you would recognize as a fish or a crab or a clam that haven't changed very much in external appearance, like general appearance. So Tully monsters, they're weird. Here's an Opabinia. There used to be arthropods that had what appear to be 5 eyes on eye stalks. That's part of the early fossil record. And so what do we do with organisms like this? The answer is we try to make those family trees, phylogenetic trees, and we're going to base them on characters. Characters are like presence or absence of some feature or some body part. You can say long proboscis, present or absent. And then if long proboscis is present, does it have this sort of clamp-like end on it? And that's, a 1 if it has, a 0 if it hasn't. And you can say, all right, eyes, how many eyes does it have? So we got 5 here. Some organisms would have 3. A lot of them have 2, because that's kind of the minimum for binocular vision, the 3-dimensional vision, rather. And some things only have 1, or have 1 compound eye, or 2 compound eyes. There's a lot of variation. But yeah, you can take any traits that exist and build a family tree out of those. And if you do it well, you can come to a reasonable phylogenetic inference. How can we approach the problem of phylogeny in weirdos like this with few surviving relatives? You compare them to other fossil things, and then you try to, like, the ultimate goal is to have fossil things on a tree, and if you can, connect that up to things that are still around, where we know about their DNA and how they relate to other animals. And that can kind of, as a proxy, let you put fossils onto a regular molecular family tree and at least see where they would have come. And big shocker, they usually come down near the base because fossils are from a long time ago. At least these fossils are. Cambrian explosion. Can fossils be used to infer behavior? You betcha. This is a coral, and we think it was a coral that was able to move. So normally corals will have an attachment point. They'll actually grow part of their body out of some other hard substance. This one, called Paleasis, never has an attachment scar on the bottom. Like it would have broken off from something, or there'd be a flat spot where it was glued down, even if it was to a little piece of algae or something. And these guys don't. And we know there are some modern corals that are able to actually pick up and use their little side tentacles to move along. And members of this group tend to have little pores, little holes all along the bottom. And you can see some of these holes connect up to holes that show up here in the cup. So this is where little fingers of the organism were reaching through and out. And the idea is, well, if they're not rooted down and they're very low and flat, like all the modern ones that are able to move themselves, and they don't have a, like they would have some of their flesh on the outside, which would appear that these did. They got a rim here that must have been built up like underneath flesh. They were quite possibly little mobile corals. Yeah, probably not fast. Yeah. But maybe they got the job done, at least for a while. Okay, how can you come up with a good hypothesis when comparing fossil to extant animals? Like here, we're comparing what appears to be red blood cells and little blood vessels from inside the exceptionally preserved leg bone of a dinosaur. Now, the proteins are all probably cross-linked, but they actually retain some of their flexibility. They said, yeah, you can bend these and they pop back into shape. Really neat. And so they've compared them here to the same things, the little femur that they've exposed to the same process, like an acid treatment to dissolve the bone. And they say, yeah, you know what, we get out little blood vessels and they basically look the same. And that makes sense because dinosaurs and birds are part of the same group, so those are pretty close to each other. You might have a more difficult comparison if you're comparing dinosaurs to turtles, a very different actual relative. Dinosaurs are not really reptiles, yeah. It might go even worse if you compare it to, I don't know, the hard parts of a clam or something like that. Generally, you want to get as close as you can, and you can figure out how to do that with your phylogenetic trees. So there you go. That's just a nice little taster to see some exceptional fossils and some of the sorts of evidence that we'll at least cover in this course. So I hope that helps.